Okay, let's get started for the KGRI lecture. And I would like to welcome you to the KGRI lecture series. It's a great pleasure and honor to have Dr. Yonehara from Denmark today, and we're going to have a great talk. So let me first introduce Dr. Yonehara, so you can sit down, briefly for his biography. He graduated from University of Tokyo in the veterinary medical science in 2003, and he moved on to a PhD career in the Sokendai in Okazaki at the Dr. Noda's lab. And from then on, actually, he has been working on the retinal circuit, and he moved on to Switzerland now to do a postdoc work at Dr. Rosker's lab. And he did a fantastic research there. And then from 2015, he got an independent position as a group leader and also associate professor at the Aarhus. Is that right? Right. I said Aarhus, and that was wrong. Aarhus. Is that right? University in Denmark. So he has been working on the retinal circuit, which is a great system for a long time now, maybe like 15 years. And he has been the leader in this field, and he has awarded for many prizes and honors. Just name a few. He got the Japan Neuroscience Society Young Investigators Award in 2015, and the Swiss Opts Award 2016, and many others. And as I said, the retinal circuit has been a great system to work on from the molecule to the cell types to the circuit to the behavior, and that's exactly what probably we're going to hear for today's talk. And his title of the talk is Visual Motion Processing, Cell Types, Circuits, and Diseases. So without further ado, please, Dr. Yonehara.
Thank you very much, Dr. Nuria, for kind introduction, and also thank you, Dr. Ishikawa, for giving me this opportunity to present our work today in Keio University. The first steps of vision occurs in the retina, which is an accessible part of the brain. The photoreceptors generate neuronal signals upon receiving visual input. Then those neuronal signals are transmitted to downstream excitatory bipolar cells and the output of retinal neurons, ganglion cells. The task of retina is to extract different visual features such as colors, edges, and different categories of motion from the scene. And those extracted features are sent to higher brain centers in parallel by the action of ganglion cells. And those excitatory pathways are modulated by inhibitory horizontal cells and macularin cells. Mouse retina has at least 40 different ganglion cell types, and at least 8 types among those 40s are dedicated to analyzing motion directions. The ganglion cell type, which is sensitive to the direction of image motion, is called direction selective cells. So these neurons show sparking activity when light stimulus moves across the receptive field of these neurons. And this sparking activity is direction selective, and the direction to which cells show a lot of sparking is called preferred direction, and the opposite direction to which cell with little activity is called null direction. In mice, there are 2 major classes of direction selective cells. One is on-off DS cells. And here, each line represents the preferred direction of individual neurons. On-off DS cells are found in 4 directions: superior, inferior, temporal, or nasal directions on the retina. And these neurons project axons to the lateral geniculate nucleus of thalamus and superior colliculus of midbrain. And in contrast, on-DS cells are found in 3 directions: superior, inferior, and temporal on the retina. And these neurons project axons to the nuclei of accessory optic system, such as nucleus of optic tract, dorsal terminal nucleus, and medial terminal nucleus. And this on DS cell pathway mediates a reflex of eye movements called optokinetic reflex, which is for gaze stabilization. This optokinetic reflex is induced by the movement of entire visual field. For example, when you are on a train looking at the scenery flowing, then this motion induces this kind of eye movement reflex, where I slowly follow the motion and quickly go back. And the directional selective cells receive glutamatergic excitatory input from bipolar cells and acetylcholinergic excitatory and GABAergic inhibitory inputs from starburst amacrine cell, which is type of inhibitory neuron, amacrine cell in the retina. Interestingly, when you do whole cell recordings from DS cells, cell body, you will see that both iPSC, inhibitory, and EPSC, excitatory inputs are both already tuned to motion directions, meaning that amplitude of iPSC is larger for null direction motion, and the amplitude of EPSC is larger for preferred direction motion. The circuit mechanism that creates directionally tuned inhibitory inputs is well studied. I will explain this in next slides. But the mechanism underlying the tuned excitatory inputs remained unknown. And EPSC should be a mixture of cholinergic and glutamatergic input. So that we have two questions here. One is glutamatergic input directionally tuned from bipolar cells, and is cholinergic input tuned from starburst amacrine cells? So we addressed these questions. Then the important circuit mechanism for creating directionally tuned inhibitory input is spatially asymmetric connectivity from starburst amacrine cells to DS cells. So here, these black cells are starburst amacrine cells. These neurons have cell body and processes radiating away from soma. Then this is DS cells. Let's say preferred direction of this DS cell is left to right. In this case, the starburst cell processes that point in the null direction of DS cells make GABAergic synapses and transmit GABAergic inhibitory signal. But processes that point in the preferred direction make less synapse. So as a result, if light moves in null direction, first this light activates the processes of starburst amacrine cells on the null side. GABA is released, so the spiking of DS cell is suppressed. This is the basic mechanism of directional selectivity in the retina. And second mechanism, which is also important, is that processes of starburst cells have centrifugal directional selectivity, meaning that GABAs are released efficiently when light moves from soma to the tip of dendrites. So when these two mechanisms are combined, DS cells achieve directional selectivity. When I was a PhD student in Okazaki, Japan, I accidentally identified transgenic mice in which only inferior sensitive type of on-DS cells are labeled with GFP on the retina. This is whole mount picture of these transgenic mice, where soma of DS cells are labeled. And we collaborated with Dr. Ishikane from Riken Brain Science Institute, and he did recordings of spiking from these cells, and he found that these cells spiking are tuned to inferior motion on the retina. So this was first demonstration of a genetic labeling of single subtype of DS cells, suggesting that different subtype of DS cells are genetically distinct. Then we found that these specific gene expression patterns were already observed from the early embryonic and postnatal stages. So we use this mouse as a tool to understand how circuits around DS cells are established during development. Then here, what we did is to label starburst macroin cells with channel rhodopsin by using chat-Curi mice in which only starburst cells are labeled with Curi recombinase and injected AAV encoding Curi-dependent channel rhodopsin. Then in this retina, DS cells are GFP labeled and the starburst cells are channel rhodopsin labeled. Then here, I did whole cell recordings from GFP cells while photostimulating surrounding starburst macroin cells to map inhibitory connectivity at different developmental stages. Then what we found is that at P6 postnatal day 6, inhibitory input is spatially symmetric. However, it becomes asymmetric at P7, 8, 9. So this development occurred before eye opening of mice, which is around P13. So this suggested that the specific genetic mechanisms underlying this development of asymmetric inhibitory connectivity. In order to search for molecular mechanisms, we focused on human genetic disease called congenital nystagmus. So this is a video of patients. This disease is characterized by two symptoms. One is horizontal oscillations of both eyes along horizontal axis. And the second symptom is lack of optokinetic reflex along horizontal axis. But vertical reflex is normal. Then 70% of this disease is caused by mutation in FRMD7 gene. But it was not known how mutation in this gene leads to this eye movement phenotype. Sandra Siegert in the lab of Boton Roska did a cell type transcriptome analysis of mouse retina, and she found that FRMD7 expression is localized to starburst macroin cells in the retina. So this is the starburst macroin cell. So we thought this may be related to the defective retinal directional selectivity. So we analyzed FRMD7 knockout mice. So we found that this is wild type mouse showing nice optokinetic reflex, so slowly following the gratings. But mutant mouse lacked this horizontal optokinetic reflex, similarly to human patients. And I don't show you data, but even these mice can show vertical reflex in response to vertical motions, similarly to humans. Then we next recorded spiking activity from retina. In wild type, we can identify on-off DS cells in 4 different directions: superior, inferior, temporal, nasal, and on DS cells in 3 directions. But in mutant mouse, we found that horizontal direction selectivity is missing, both in on-off DS cells and on DS cells. Then there are 2 possibilities to explain this lack of horizontal direction selectivity. One possibility is that cells, horizontally tuned cells are dead and lost. Second possibility is that cells are there but lost tuning, becoming non-direction selective. To test these possibilities, we performed two-photon targeted patch clamp recordings from DS cells, which is labeled by GFP in HOXD10 GFP mice. In wild type, cells are tuned to some of the directions, the spiking, high frequency spikings in some of the directions. This is preferred direction. And amplitude of inhibition is larger for null directions and smaller for preferred directions. So this is wild type. In FRMD7 mutant retinas, what we found is that the spiking becomes non-direction selective. And also the amplitude of inhibitory input, iPSC, becomes non-direction selective. So these results suggested that FRMD7 is a master gene for establishing spatially asymmetric inhibitory circuit from starburst macroin cells to DS cells. And likely humans really rely on retinal direction selective circuit for mediating optokinetic reflex. So these are kind of introductions. Then from here, I'd like to talk about 3 topics which are ongoing in my lab in Denmark. The first topic is development of spatially asymmetric inhibitory connectivity. So spatial asymmetry in neuronal circuits play an important role in creating basic building blocks of neuronal processing. So well-known examples are like visual cortex direction selective cells and DS cells in the retina. But the molecular mechanisms that underlie the establishment of asymmetric circuits remained unknown. So we addressed this question by continuing working on FRMD7 molecule. So the FRMD7 has a so-called firm domain in the N-terminal. Then this domain is believed to be important for interacting with the plasma membrane. And many of the human mutations are concentrated around these firm domains. But we don't know the function of this protein yet. We modeled this protein structure, and actually we found that these firm domain regions are well conserved across species, and this part is positively charged, indicating interaction with the plasma membrane. Then immunohistochemistry suggests that really this protein is localized to chat-positive starburst macroin cells from P3 through P7. And in order to understand the interaction molecules of FRMD7, we performed mass spectrometry and found some candidate interaction molecules. But we don't know yet which may be important signaling. So now we are trying to knock down different molecules, interaction molecules, to see effect on retinal motion computation. And in order to search for more relevant proteins, we have been performing 4-step screenings. First is cell type transcriptome, actually developmental cell type transcriptome. So we prepared different transgenic animals where single cell types, ganglion cell types, and different macroin cells are labeled with fluorescence proteins and sorted with a fax. Then performed RNA sequencing to identify cell type enriched molecules. Then we brought in mutant mouse for genes enriched in starburst macroin cells, which was our focus this time. Then measured optomotor reflex. We use this reflex because we found that this reflex, the optomotor reflex, is a movement of head rather than eyes. So mouse use more heads to follow stimulus when head is not fixed. So we use this reflex to assess the function of retinal direction selectivity. If we found some phenotype, then we isolate retinas and put on microelectrode array, which has 26,000 electrodes, which is high density, high throughput. Then finally, to understand synaptic mechanisms, we perform two-photon targeted patch clamp recordings from DS cells in this gene knockout backgrounds. So the second topic is computation of motion speed and direction in the retina. So the understanding of the retinal direction selective circuit has been biased to direction selectivity mechanism rather than speed selectivity, even though it's known that different direction selective cell classes are tuned to different speeds. For example, some motion speeds are fast and some are slow. And it's important to detect the speed of motion for animals. For example, animals show optokinetic reflex to slow speed, but not to fast speed. And they have different meanings for the animals. And on-DS cells are known to be tuned to very slow speed, which in mice, with optimal speed is around 150 micron per second on the retina, which corresponds to 5 to 6 degrees per second. On the other hand, on-DS cells have a broad speed tuning, so the spike generated in response to broad velocity of stimulus. But it's been unknown what kind of circuit mechanisms create such speed tuning differences between different cell classes. So we addressed this question. Then there is a model which we focused, which is called Hassenstein-Reichard motion detector model. This was proposed by German physicist Reichard and biologist Hassenstein. This model requires two receptors converging onto postsynaptic neuron, and one signal transmission line has a time delay. Only with these components, you can create selectivity to both speed and direction. So now I move this light stimulus from left to right. Then first this receptor is stimulated and signal propagates, followed by stimulation of this receptor. Then at certain speed, when the time difference of this activation of these two receptors corresponds to this delay, this input can arrive simultaneously in postsynaptic neuron. Then you have a nice summation, high amplitude of the input. But if light moves in opposite direction with the same speed, then obviously this signal arrives earlier than this signal. So the summation is less efficient compared to this. With this kind of mechanism, direction selective cells could create tuning to speed and direction. Speed, so because if this light moves in different, if too fast or too slow, would degrade this summation. So if in order for this kind of a mechanism to operate in retina, you would need different bipolar cell types with different temporal dynamics, like slow bipolar cell input and fast bipolar cell input. So we tested this hypothesis by two-photon glutamate imaging. So we targeted IGL sniffer, which is a glutamate sensor, to the dendrites of on-DS cells by using PCDH9 Cre mice in which only on-DS cells are labeled, and we presented this kind of a modulating spot light stimulus, which changes luminance dynamically to map the temporal dynamics of glutamate release onto different parts of dendrites. With this method, we found many different glutamate release from bipolar cells with different temporal kinetics. Then by clustering analysis, we found that there are at least 6 distinct glutamate release types based on temporal dynamics. Some are fast and transient, some are very slow glutamate release. Then in another experiments, we used the dense noise stimulus to map temporal and spatial receptive field of individual glutamate release. Then interestingly, we mapped these glutamate release groups onto the dendrites of single on-DS cells. And we found that fast inputs are biased to null side of dendrites, and slow inputs are more biased to preferred side dendrites. So these pink ones are fast transient, and red one fast sustained, medium brown, and slow transient blue, slow sustained dark blue, and very slow yellow. And this is a histogram of distribution of different glutamate release types along motion preference axis. So kind of this is reminiscent of Reichard motion detector. So slow input and fast inputs are spatially segregated. So it's probably easy to imagine that this kind of input structure can produce Reichard detector-like motion, speed, direction, computation mechanisms. Then in order to determine functional structural correlation, we collaborated with Professor Kevin Bregman, Caesar in Bonn, Germany, which is part of Max Planck Institute. And he traced bipolar cells from dendrites of on-DS cells with a 3D electron microscopy. Then I skipped details, but he found that this some bipolar cell types, namely 5. So before that, there are at least 3 different type 5 bipolar cells, type 5T, O, 5I, in the mouse retina, which is a kind of axon terminals are stratified at similar depth in the inner plexiform layer of retina. Then he found that 5O, 5Is are more biased to null side dendrites. On the other hand, 5T is more biased to preferred side. Then he got this kind of histogram. And by comparing this glutamate imaging-based histogram and electron microscopy-based histogram, we can infer which anatomical type may correspond to which release types. Then this is summary of input glutamatergic bipolar cells or onto the dendrites of on-DS cells. So we found that type 5, group 5 is more biased to the far preferred side and followed by group 6, which we think are glutamatergic amacrine cells. And groups 3, 4, 2, 1. I didn't show you data of pharmacology, but we found that some such transient glutamate release types cells receive presynaptic GABAergic inhibition. If we block GABAergic circuits, then this release type became more sustained. Then very fast bipolar cell types were sensitive to TTX, suggesting that these bipolar cells express voltage-dependent sodium channels and spikes. I forgot to mention, but usually bipolar cells are not spiking neurons, but it's reported that some bipolar cells are spiking neurons. So we think these first ones are actually spiking. The anatomy suggests that this 5T, V-glut T3 positive amacrine cell, 3, 5T, 5I, 5O. And here what is interesting is that both glutamate release group 4 and 5 were both assigned to type 5T bipolar cells. So this suggests that axon terminal of bipolar cells receive GABAergic presynaptic inhibition if these synapses are made on more null side of dendrites. So there is a kind of a location-dependent presynaptic inhibition. This was suggested from our study. Then next we created a model, linear filter model, we call delay and summate model. So we spatially distributed response units based on experimental data. Then in response to 200 micron per second slow speed, each line indicates input from different units or bipolar cells. Then in response to this kind of slow speed, this input arrives almost like in same time in time. This is time axis. Then achieving high summation, high peak. But for null direction, these inputs arrive in different timings, so the summation is worse than this direction. So this is just to show that this model was able to create this tuning to slow speed. Similarly to experimental data, this is this purple area. Okay. Then very interestingly, we found that so this I don't explain details, but this space-time receptive field of EPSC was determined by EPSC recordings in response to dense node stimulus. This indicates that there's slopes, indicating that the spatial temporal receptive field is kind of diagonally tilted on these cells. And we found that this tilt is impaired in FR-medicine mutant mouse. So this indicates that sustainedness transients of gradients in glutamatergic input is kind of reduced in FR-medicine mutant mouse. So probably this is why FR-medicine mouse couldn't show optokinetic reflex. And it's very intriguing that how this phenotype arises in this mutant, because FR-MID7 expression is localized to starburst macroglin cells, and it's known that starburst macroglin cells don't make synapse with bipolar cells. So this is our future question. Then our computer simulation predicted that by modulating GABAergic signaling in different bipolar cell types, for example, by making some bipolar cells more sustained and some bipolar cells more transient by modulating GABAergic receptor signaling in FR-MID7 mutant mouse, we could improve directional selectivity in these FR-MID7 mutant retinas. So we want to design genetic repair for directional selectivity in congenital nystagmus retina in future. Then I mentioned earlier that also acetylcholine is released from starburst macroglin cells to DS cells. And it's not known whether these are tuned to motion direction or not. Then we used the acetylcholine sensor called GAC, developed by Yulon Lee in Beijing University, and targeted to one of DS cells, dendrites, and did two-photon imagings to see how cholinergic releases are tuned or not. Then I skip details, but what we found is that individual inputs are tuned to motion direction, but as a whole, the tuned directions are multi-directed or random. And we added this ambenonium to block acetylcholine esterase. So I forgot to mention the question. So one question here was, it was not known whether cholinergic transmission is synaptic or volume transmission in the retina. So to test this idea, we forced cholinergic transmission to become volume transmission by blocking acetylcholine esterase. Then this resulted in less tunings. Yeah, so indicating that in normal condition, cholinergic transmission is more synaptic rather than volume transmission. And the tuning was random. Okay. Then Gautam Avtramani group in Victoria is doing electrophysiological recordings, and our group is doing imaging. Then we are combining this data to write a paper. The last aim is role of retinal motion processing in cortical computation. So as probably you know, visual cortex have direction selective cells. There are neurons which are sensitive to the direction of image motion. And also retina has direction selective cells. So then what is the relationship of retinal direction selective cells and cortical direction selective cells? So retinal circuits extract like 40 retinal movies, then send this in parallel to brain visual centers. Then our question was, what will happen to cortical motion computation if we knock out retinal motion computation? So we used FR-MID7 mutant mouse as a tool to knock out retinal direction selectivity to address this question. Then in primate, so the signals departed to retina is transmitted to LGN primary visual cortex V2 and to different visual areas, higher visual areas. And some pathways are more maybe associated with space action functions, and some pathways are more associated with object recognition. And the recent studies identified also higher visual areas in mouse visual cortex after primary visual cortex. But it's still not known what is the relationship between primate and mouse higher areas, and how these response properties in different higher areas are generated by circuits. So we performed intrinsic imaging on control and FRMD7 mutant mouse to map higher visual areas in addition to primary visual cortex. Then we didn't see difference in the areal size and positions between these two genetic backgrounds. Then we presented motion stimulus moving in different 4 directions, posterior, anterior, dorsal, ventral, to different force speeds, 10 to 60 degrees per second from different areas, primary visual cortex, LM, AL, RL, AM, PM. And we found that response to posterior and anterior motions was significantly reduced in area RL, rostrolateral, but not obvious in other areas, such as PM or AL. In addition to, of course, and also primary visual cortex was affected. And next we performed two-photon calcium imaging to look into responses of single neurons in different brain areas, but we focused on RL and PM because which didn't show any effect. Then again, we found that the response to posterior is the most strikingly reduced in FRMD7 mutant mouse. It's not obvious here, but we found that these many cells become to prefer a slower speed in this mutant mouse. Then it's known that primary visual cortical neurons in layer 2-3 make target-specific projection, meaning that the response property of neurons projecting to certain area is similar to the response property of target areas. So one possibility was that the sensitivity to retinal direction selectivity is already created at the level of primary visual cortex. So we made a retro AAV encoding GCAMP and injected to either PM or RL and retrograde relabeled layer 2-3 neurons in V1 and did calcium imaging in control and FRMD7 mutant mouse. And again, we found that the PM projecting cells are not really affected in terms of a fraction of DS cells, but RL projecting cells have reduced fraction of DS cells. And PM projecting cells prefer more slower speed, but this was not affected. But RL projecting neurons prefer higher speeds in control mouse, but in mutant mouse, many cells become to prefer slower speeds. So the PM is not really affected, but RL has major effects. So these results suggested a segregated cortical processing stream for signals from retinal DS cells. So here, on-off DS cell pathways indicated by blue, and non-DS cells pathways are indicated by magenta. So it's already demonstrated by Marshall et al. And Cruz-Martin et al. That these DS cells project axons to the shell area of LGN. And these neurons make axon projection to layer 1, 2, 3 superficial, not to layer 4. Then indeed, we found that layer 4 responses were not altered in FRMD7 mutant mouse. So likely, these DS cell signals are skipping layer 4 and directly reaching to layer 2, 3. And there exists a specific routing mechanism that pass this signal from DS cells to RL, but not to PM. So the next intriguing question is, what is the functional role of rosterolateral? And what is the counterpart of primate cortical areas? So our hypothesis is that processing of rotational and translational optic flow in primate, MT, MSTD, VIP, ventral, intraparietal cortex relies on retinal DSL. So the subtitle reported that the preferred directions of DSLs on the mouse retina is aligned to the axis of optic flow. When mouse translate, like move forward, it creates optic flow from forward to backward. Then these preferred narrow axis of DSLs are actually aligned to these flow directions, indicating that all of the DSLs are adapted to analyze optic flow, which is induced by kind of movements of mouse, locomotion of mouse. Then Tedvilla Daisy reported one of the DSLs in macaque retina in this year's ARVO. So it will be very interesting to test whether direction selectivity in these areas in primate, monkeys, and humans may rely on this signal from one of the DSLs, similarly to humans. There are other anatomical functional evidences that supporting an idea that RL may correspond to these areas. So there are three future directions in my lab. So first, it's circuit asymmetry in the retina. So we continue to use asymmetric inhibitory circuits and asymmetric excitatory circuits, which we discovered recently, as a model to understand how spatial asymmetry in neuronal circuits arises during development by different genetic and perhaps activity-dependent mechanisms. The second is cortical processing of optic flow for spatial navigation. So there are neurons in the brain which represent spatial information such as place cells, grid cells, head direction cells. Then it's already suggested that the visual input contributes to the receptive field of those space-related cells. And our hypothesis is that optic flow detected by retinal DS cells and concentrated in rosterolateral in visual cortex may contribute to the response property of these neurons. So we want to test this hypothesis by performing imaging from freely moving mice or head-fixed mice in virtual reality conditions. So with this project, we want to understand how visual processing contributes to spatial cognition. The last is a new project for us, which is restoring vision by in vivo direct reprogramming. So here our aim is to treat glaucoma, where ganglion cells are degenerated and gone. Our strategy is to create ganglion cells de novo from mural glias by putting different genetic and epigenetic factors. So this is last slide. So I'd like to thank people. So this is people in my lab. Like many Danish students and international postdocs. And the collaborators, Kevin Bergman did electron microscopy, Gautam Awatramani is doing electrophysiology for cholinergic transmission project. And now we are also working on bipolar to on-off DSL circuits. For this, David Bass and Shai Sabaz are tracing electron microscopy. And we collaborate with Boton Roska and Jacek Kroll in Basel to understand the FRMD7 protein's function. And Paul Nissen do a structural analysis to understand the structure of FRMD7 protein. And Dudak Vichani for behavior studies, Ben Honore for proteomics, Karl Klaas Konzelman for rabies development, and funding. So our institute was founded by Lundbeck Foundation and Oxford University. And I received the ERC starting grant from EU and many funding agencies in Denmark. Thank you very much for your attention.
Thank you very much for the beautiful data and the fascinating talks. And any questions and comments from the floor?
So you suggest that functional role of on-off DS cell is they are called the optic flow. So the speed tuning of on-off DS cell is bandpass type. The locomotion speed and the sensitivity of on-off direction selectivity cell is matched or not? Tuning boundaries.
Sensitivity of on-off DS cells and also
speed sensitivity is matched to the moving locomotion speed.
Yes. So we looked into the optic flow speed when mouse is locomoting in different modes. So we found that when mouse start to slowly run, then on DSL spiking is the most robust. But what we think is most relevant is the optokinetic reflex. So our hypothesis is that when mouse is doing optokinetic reflex, so our hypothesis is that on-off DS cells, the signals are transmitted to cortical areas when mouse is not doing optokinetic reflex. So when mouse is very slowly moving, reflex occurs and try to fix it. But above this threshold, when mouse starts to walk, then mouse's visual system switch to analyze motion, optic flow. This is our hypothesis. And actually, speed tuning matches with this hypothesis. Yes.
Thank you.
Any other questions? Comments? Yes, please.
In humans, central nucleus of horizontal nystagmus is PPLF, and on the other hand, the central nucleus of the vertical nystagmus is called RI, MLF. So it's a different location, it's different central nucleus of the thalamus is the central nucleus of the central neurons controlling nystagmus. So my question is that the only gene could change the sensing of vertical motion, vertical movement, or they also could change the projection pattern, or.
We looked into projection patterns of axons, but we didn't see any change in these mutant mice. So I'm talking about the projection from retina DSLs to central areas. Yeah, this was not really changed. Yeah, but actually I didn't tell, but FRMD7 protein expression is also observed in brain areas. Not visual cortex, visual areas, but vestibular nuclei and some oculomotor nuclei. It's possible that these expressions in these areas may relate to nystagmus. We have to see this. But at least what we suggest is that the lack of optokinetic reflex can be explained by retinal circuits.
Any other questions, comments?
The layer 4 neurons in visual cortex has direction selectivity?
Yes, they have.
And my question is, on layer 2-3, direction selective neurons are affected by layer 4 direction selectivity. And also from retinal direction selectivity or only from retinal direction?
Right. So what we think is that when motion is slow, these cells are impacted both by layer 4 direction selectivity and maybe less retinal direction selectivity. But when motion speed becomes high, this, like what I believe is that this thalamocortical synapses at layer 4 may not be able to create direction selectivity, then, or at least input from retinal DS cells become more dominant to override direction selectivity of this. So actually, I didn't show you, but we found that many neurons in layer 2-3 change preferred direction. When motion is slow, many cells are tuned to many different directions. But when motion becomes fast, this preferred direction shifted to posterior. So this indicates that when motion is slow, it's more driven by layer 4 mechanisms. But when motion becomes fast, retinal direction selectivity signals become more dominant to bias preference to posterior to analyze kind of optic flow. So yeah, this is our hypothesis.
Yeah, it's quite interesting. Thank you.
Yeah.
Mean that the direction selectivity change depending on the temporal frequency or speed?
Temporal frequency. Actually, right. So in our analysis, we fixed the spatial frequency, and we just changed the temporal frequency. In this condition, we found that they shift depending on temporal frequency. But it's a bit complicated, so there are evidence that retinal on-off DS cells are more tuned to speed rather than spatial temporal frequency. So layer 4 mechanisms are likely more tuned to temporal spatial frequency. But so they start to interact. It's a very complex thing. But I agree that we have to systematically change spatial temporal frequency. Yes, we are actually doing this now.
Actually, one quick question from me. So syndecan 2, the name suggests there's different types of syndecans. So I wonder if, and then that may contribute to the coding of the circuit. And I wonder if there is similar proteins or the genes to the FMRD, FRMD that may contribute to the different circuits in the retina or other areas. Is that known?
So there are 8 FRMD7, from 1 to 8. And some FRMD7s, no, sorry, not 7, FRMDs, some like, I forgot which, 3 or 6, involved in polarity of cells in, I thought the intestine or something, to kind of basal apical polarity pathways. I don't remember details, but as far as I know, it's not known other FRMD7s involved in neuronal circuits functions or development. So the 7 is, I think, first one implicated. And in retina, I think FRMD7 was the only FRMD which showed interesting gene expression pattern in cell types. And syndecan has 4, from 1 to 4. And actually 4, I thought 4 is also expressed in starburst macular cells, but very surprising that only knockout of 2 created this phenotype. And indeed, there are evidences that different syndecans have very distinct functions. So, yeah.
Thanks. Okay, any other questions, comments? It's crystal clear. No? Okay, so it's about time, so let's thank the speaker again.